大家好，脸书上的朋友，大家的饮料、零食、耳机还有键盘都准备好了吗？我们今天是要来做这个虚幻生命展的一个前期的直播哦。那我是曹存慧，我是今天的直播的主持人。然后等一下如果有任何问题的话，请大家可以留言在脸书底下的留言区。然后今天我们很高兴有邀请到这个Paul VANOUSE 来我们的节目当中，然后跟我们在线上对谈。那等一下他就会出现在画面上了。他现在人还在纽约州，然后他是这个纽约州立大学水牛城分校的教授，同时也是一位艺术家。那他过去几年常常在各国展出，然后台湾大家很熟悉的这个林兹电子艺术节，他也曾经得过几次重要的奖项哦。那他除了Paul 除了在纽约州立大学水牛城分校教书和创作之外，也负责运作那边的一个 Collapse 生物艺术中心，也许可以翻译成联合或者是复合生物艺术中心。然后他同时也共同跟其他的教授共同负责那边的一个Master of Fine Arts 艺术硕士的运作，然后那个艺术硕士的学位的名称叫做 Emergent Practices， 可能可以翻作是正在发生中的艺术实践吗？等一下可以跟他多聊一些这方面。然后他同时在明年初的时候也会有一件在林兹得奖的作品叫做 Labor， 这次我们翻译成劳力，会到空总的C-Lab， 可能在图书馆这边会做一个展演。那等一下我们就邀请他来跟我们分享他的作品。Paul, do you want to say hi to the audiences?
Yes, hello. Hello, everybody. And thanks to you, Teresa, and Pingy, and Aaron also. I wish I could be there in Taipei now. That was my plan. Not all plans work out.
We are looking forward to seeing you next year. So I just introduced you as an artist. Do you identify yourself as a bio artist?
Yes. More broadly, I think of myself as someone that works in emerging media forms.
Uh-huh.
You know, and addressing those the issues raised by the technosciences through them as forms in and of themselves. So I use biotechnology to speak to biotechnology. Does that make sense?
Yes. So do you want to talk to us more about your recent works?
Yes. Okay. Well, I should say that I've been using the emerging technosciences as medias for artistic expression now for my entire career, and for the last 20 years I've worked with DNA as a medium for and molecular biology protocols as artistic techniques. So in this talk, I thought I'd begin with a few of the earlier works and then move on to my latest scent-based work, Labor. And finally, I thought I'd conclude by showing you my lab Coalesce, which I started at the University of Buffalo about 6 years ago. Mm-hmm. I wanted to actually present from Coalesce, but the coronavirus is ravaging the U.S., and it's mostly because U.S. citizens are receiving such mixed messages from our president, for instance, about taking it seriously. And so in this context, I present from home really to patriotically social distance, because Black Lives Matter and science is real. And so I really only use the lab now as it's definitely needed for my work. Most of this work I've taken on misleading metaphors and hype surrounding DNA and the Human Genome Project, but as a critique unified against also an overly reductive worldview. And this is a worldview, of course, which has been central to Western modernism. And just in case this term reductivism is too vague, really what I mean is when we try to reduce something complex and multidimensional to a single cause, when we oversimplify. This is the basis for this talk today. My biotechnological work began about 20 years ago, and it really stemmed from a round table in Seattle, Washington. And at that round table, some of the most important genome scientists of the time were together to discuss the implications of the Human Genome Project. There was 6 artists and 6 scientists. Just to refresh people, in case they don't remember, the goal of the Human Genome Project was to map every single gene in human DNA. There are about 30,000, and there are about 3 billion base pairs. These things, these A, C, Gs, and Ts, that are the way we see DNA when we represent it as a code. There was all kinds of hype and misleading metaphor around that project, particularly at this time. So I'll begin with that moment 20 years ago, looking at, the future of DNA in society. Some of the most problematic hype for me of the time was the misleading metaphors surrounding DNA. So things like the idea of DNA as being destiny, DNA as the code of codes, and even what's hard to recognize as a metaphor, the term DNA fingerprint. And the metaphors for me were problematic for a few reasons. One was their fatalistic determinism, the idea that we are only our DNA. And secondly was the emphasis that they place upon the idea that DNA is a code rather than a material substance in every one of our cells. Also the idea that DNA is somehow the master molecule that drives the rest of our cells and they are more important than our environment, etc. All of these metaphors I saw as oversimplifying, but also misleading and self-aggrandizing, like really just seeming to be out for their own purpose. So with my work, I've tried to complicate the fixity and the wholeness, ideas of essence and determinism in favor of what I call dynamic change. Terms like hybridity, multiplicity, uncertainty, complexity. But I'm also undermining this idea in the work that life is information, which was especially strong in the early years of the 20th century. I really hope everybody can hear me okay. It's strange. It's strange being so far away and literally just looking at a screen.
We can hear you fine.
Great. Key to all the work I've done has been to use the tools of visual studies to understand scientific artifacts as cultural artifacts and to trace the social processes through which they came to be accepted as facts, right? Many of these projects use the analytical tool gel electrophoresis, which is the image you're looking at here, as an expressive medium. The images created by this technique are often referred to as DNA fingerprints. In my work, I'm always emphasizing the idea that DNA is malleable material. Here's how that process, DNA fingerprinting, works. If you look at A in the diagram, you can see a bunch of DNA fragments of different sizes, 2 different sizes. If I were to take those fragments and to squirt them into that rectangular opening at the bottom of the gelatin, and then apply voltage to that gelatin, you would see by letter C that the DNA will have moved in the gelatin at a rate that's inversely proportional to its size, which simply means in a DNA gel, you squirt DNA into a spot, you apply voltage, and the smaller pieces of DNA move the fastest, and the larger pieces move slower. And if those pieces are identical in length, they will move at the exact same rate. And that's all that DNA fingerprinting is about through this process. One of my greatest concerns about the Genome Project when I started this work was the way which race would be addressed. In 2000, following the completion of the rough draft of the Human Genome Project, a team of UNESCO scientists optimistically stated there is no scientific basis for race. This was a concern because the Human Genome Project marked a return to an interest in human genetics not seen since the eugenics movement. The original title for the Human Genome Project was neo-eugenics. And it was only suggested by the Nobel laureate James Watson to change the name because he said, well, people will have bad recollection of eugenics, which caused the death and sterilization and associated with genocides all over the world. So the name was changed. And I think many people breathed a sigh of relief. But I assumed that racism could exist even if race did not. That it would simply leave the body's surface and go molecular. That now, rather than declare a racially stigmatized body as genetically unfit, genes common in that stigmatized population could then be declared as unfit. This is what I call molecular racism. I recalled the book Race Crossing in Jamaica, written by an American eugenicist, Charles B. Davenport, in 1921. The book described black and white racial hybrids as less than the sum of their parts. This struck a chord for me because my biological family members are hybrid of Jamaican descent. So having a black mother and a white father, my identity was always really complicated. In the US, sometimes I was black, sometimes I was white. Many times I was assumed brown or Asian or Latino, though I'm not descended of any Spanish speakers. So in my first large-scale DNA work, started in 2000, I wanted to take on this idea that technologies can highlight minute differences, often at the expense of greater similarities, and that these differences are always value-laden. As a mixed person, I grew up wondering, why is it my skin was lighter than my sister's, who is shown in this slide here. In many ways, the colonial obsession with difference is internalized in post-colonial subjects like me. So in a form of historical condensation of eugenics and identity, I wanted to construct a dramatic experimental system to finally determine who in the family, which skin color is the most genetically fit. How better, I thought, than first extracting individual skin color genes, DNA, from each family member, and then taking this DNA and literally racing it against each other. in a DNA gel. And I proposed this project for the first US exhibition of bioart called Genesis, which took place at the University of Washington in 2002. They commissioned the project and they premiered it there in 2002. And I collaborated with a remarkable biologist known as Mary Claire King, who did all the DNA work at the time in the lab. In the project, I used the current medium of genomics, DNA fingerprinting, as my artistic medium. So I literally inserted the family skin color genes into unique lanes of the DNA gel. I don't know if you can see in the slide, there are tiny rectangular holes that I'm squirting the DNA in here. So first mother, then father, then sister, then brother, who was me. And then wanted to see which ones would move the fastest. The humorous irony here is that this would be a method of testing our genetic fitness. So the whole piece is kind of a pun on this whole notion of genetic fitness. The skin color genes can be raced. And in this case, it was literally a race about race, in which our actual bodies were now erased. And what I was trying to do in the piece was demonstrate race was actually a verb. That subjects can be actively raced or racialized, that we're not born into race. Rather, it's a machinic operation. And this operation is like an assembly process that draws on all kinds of sources and forces, scientific literature, poetic metaphor, state policies, national boundaries, economic structures, etc. The inscription device here then created visual evidence of race. It's very complicated. I'll show you, I hope this works. I hope you can see this. So the race is literally captured by video camera and computers, which keeps track of the position. Since there are multiple genes responsible for skin color, I needed to keep track of the exact data about each one. Is the sound on that okay? Was it too loud? Just to make sure.
It's okay.
Okay, good. So this was an admittedly pessimistic view that new technologies would be used to reinforce familiar stereotypes. But James Watson, again, the Nobel laureate of 1952, gave a lecture at UC Berkeley basically the same month that I completed the work. It was very controversial, but it also, I guess, evidenced my greatest fears of the new genomics. James Watson described an experiment by scientists in Arizona in which white college students were injected with melanin, which is the substance which makes our skin dark, and only to observe an unusual side effect. The men, in this case, supposedly developed these sustained and unprovoked erections. He said this is better than better than Viagra, because you don't even have to think about sex. That's why you have Latin lovers, he said. You've never heard of an English lover, only an English patient. So this was, on the one hand, very funny in a way, but also it showed us that the Nobel laureate himself tells us that it's not black people that can't control their libido. It's rather this molecule melanin, this blackness itself that's to blame. Indeed, it's not just the black body that's prone to promiscuity, but blackness itself. Basically, Watson here brings up one of the oldest stereotypes and tries to scientize it, tries to give it a sort of scientific veneer. So I realized that this work I was doing was very tied up in discussions of identity of the era, and I kept looking after that piece was finished at this very thing, this DNA fingerprint. Why was it called a DNA fingerprint? It wasn't from a finger. And I realized, well, okay, it's a metaphor to an earlier identification technology. And fingerprinting, like from an actual finger, has a really curious history that the metaphor carries with it. It was first used in colonial India by William Herschel, initially to prevent fraud on contracts and deeds. He later fingerprinted prisoners upon sentencing. And to understand the context of colonial India at the time, this was just after the Colonial Tribes Act was passed, which basically mapped onto the existing Indian caste system policies that would deepen notions of race, class, and criminality as linked and inherited and predetermined. This is the scene there. Fingerprinting then was further explored by Francis Galton, who was a cousin of Darwin, and also the father of eugenics. He literally coined the term in the late 1890s as a way of discerning. He was interested in fingerprinting as a way of discerning markers of heredity, ethnicity, and race, as well as to establish a national identity. Much like DNA is today, fingerprinting was imagined by many to hold all the hidden keys to the secrets of human identity, propensity, and background. To Galton's dismay, however, there is no relationship between the fingerprint patterns and race. They just have merely an indexable value. Jumping back now to the present, though, law enforcement really loves the metaphor of DNA. The director of one of the FBI directors stated that the term DNA fingerprinting invokes in the mind of a jury that we're identifying one individual to the exclusion of all others. And I thought, wow, fascinating that this technology is loved by law enforcement because it invokes something. It's almost like law enforcement loved the poetry of that term. Similarly, Sir Alec Jeffreys, who invented the technology, said, if I had called it something different, in this case, Southern blot profiling, nobody would have taken a blind bit of notice. But called it DNA fingerprinting, and the penny dropped. Literally, every crime lab all of a sudden needed to have it. It brings to mind one of my favorite cartoons from science and technology studies theorist Bruno Latour, who in his book Science in Action describes two faces of science. To the left, the old face of science we learn about in textbooks says, once the machine works, the people will be convinced. And on the other face, the younger face says, the machine will work when all the relevant people are convinced. This is a very kind of like social constructivist understanding of how facts are constructed. So in the next piece I'll show is a piece called Latent Figure Protocol. I took advantage of all the different ways that DNA could actually be chopped up. And this is a picture of a bunch, probably 50 of the 100 possibly available chemical enzymes, which can be used to chop DNA into pieces. Literally, if you were to combine these together, you can chop bits of DNA into hundreds of thousands of different patterns. Here's how it typically works. Like, I'll explain quickly. Like, for instance, if you had a piece of circular DNA called PET11A, and you added two enzymes to that, in this case, BAMH1 and APOL1, that piece of DNA would be cut in two pieces And then if you were to take those two pieces, those two different size pieces, and stick them in a gel, you would get two different bands, which you probably all now know that the little ones would move the fastest in that gel, the smaller pieces of DNA. Anyways, what I realized was that perhaps I could do molecular biology in reverse. It seems since the DNA of most organisms are known, and since every enzyme cuts like a specific sequence of DNA, I should be able to write a computer program, which is what's shown here, in which I can put in an ideal image of what I wanted to make and have this program generate the best recipe to make it. I wanted to make pictures of DNA. What I wanted to do was to turn the authoritarian essence, the DNA fingerprint, into a purely cultural image. And when I do these performances, I do this live. I create artistic representations. I wear all black, like an artist, because I didn't want the sort of authority of science here. I wanted I wanted people to be a little distrustful, like they are of artists dressed in black. The images would then be formed in the gelatin over the course of the performance, which was about an hour long. And then when they were complete, they would just slowly dissolve over the period of about a week. Basically, the DNA would diffuse out of the gelatin and just be left with this kind of sea of nothing. In all the images, I would eventually photograph the gels. And when I photographed them, I'd also print at the bottom of the photographs the actual recipes used to make each image. So you can see those little codes that kind of at the bottom of each picture show the enzymes used in each laying of that DNA gel. And I did this for two reasons. One was because I wanted to basically offer a scientific proof to show that the DNA image was in fact cultural. Like I could just literally make them look like whatever I wanted in a lab, because these are all the same organisms. And I also felt like it was almost like a source of like an open source image. Like now anybody can make this who wants to. So I've done related projects to latent figure protocol. Actually, two. The first was a piece where I again really kind of think about DNA and its relationship to identity. And in this case, it's called Suspect Conversion Center. I had a kind of painfully long, several week durational experiment with a collaborator named Carrie Sheehan. And we basically extracted my DNA from my spit and proceeded to use my DNA, cutting it to exactly the right sizes, to eventually remake the evidence of the O.J. Simpson trial in the 1990s, but using only my own DNA. I'm not going to go deeply into that piece, but the idea was I would become, my DNA would take the exact same pattern as O.J. Simpson's, as also his victim, Nicole Brown Simpson. So kind of portraying myself as all the suspects and victims of the crimes. I did this because while DNA fingerprinting is often seen as a technology to differentiate people, I believe that the best critique of this was to highlight humankind's incredible sameness. My DNA was also representing all suspects and victims. So why is it that we're always told that it's our DNA that makes us different when 99% of our DNA is the same? DNA imaging relies on knowing the very few areas that actually differ between people. And I wanted to show that we're all basically the same. So my final DNA imaging project I'll show is the America Project, in which I create DNA images of power. In this case, the US flag and the European crown. The object in the center I call the biomatter anonymizer. Audience members, in this case, empty cups of their own spit in the top. And the massive centrifuge below, I extract the promiscuous commingled DNA. I realized that since most of our DNA is identical, I can create completely stable images, even with hundreds of different human samples all mixed together. The project was really the culmination of my artistic manifesto of radical sameness. Our DNA is mostly identical. I do all the processing again live in the space. I'll show a minute of this. This is what the this is how the image actually sort of grows. And I'll say that with all these DNA projects, I work against the idea that the DNA is natural, and also that the DNA image as an imprint, when it's actually this temporal process that's often just stopped at some point. So here's a bit of how that flag image unfolds. It's very slow. And this is, it was the hardest image to make because to try to make these long smears in the DNA gel is usually what biologists try to avoid. So in this case, my scientific collaborator and I, Solon Morse, we had fun actually we actually read the troubleshooting section of a PCR manual to try to figure out how to produce the very things that most people are trying to avoid, which are these long, indistinct smears. That's the America Project. So all that DNA work reacted against the idea that DNA fully denotes who we are, what we will be, our strengths and our weaknesses, our genetic destiny. I thought it was a gross oversimplification of the complexity of life. And since 2007, though, human identity has become increasingly complicated in a refreshing way by research into the human microbiome. This research finds that bacterial cells in and around our body vastly outnumber human cells. And furthermore, the skin bacteria of even our lower arm have this like 70% variance in the strains between individuals. So in this context, then, the idea of what makes us human was again under the microscope. And not just for scientists, but for the culture at large. For centuries, we've been debating who gets to be considered a person and when. It's a question that dominates political discourse of the last few centuries because its connection to both labor and liberation in the post-Renaissance world. So how do we define human beings now? To further dethrone any conception of self, most people think the way they smell is part of their own natural condition. Put simply, your smell is like your smell. But in fact, it's more complex. It's actually a cocktail of microorganisms that live on and in us that are the culprits that determine this rather distinctive human scent. So, for instance, species like Staphylococcus produce isovaleric acid to produce a very mild scent. Propionibacterium metabolize more complex excretions from apocrine glands and responsible for vinegar and acrid odors, etc. Corynebacteria is another one. However, all these microbes act in these complex communalistic ways in the human body and utilize varied ways of metabolizing that produce an aromatic bouquet of a complexity far greater than the sum of its parts. The smell of sweat is literally the smell of just a few species of bacteria that feed upon the excretions of the human body to produce the familiar acrid and sour scents. In this sense, then, the smell of exertion is not actually human scent, unless we're willing to redefine what constitutes human. Scholars like Elizabeth Gross and Rosie Bradati and Monica Baca have sought to recast humans as more of like a structural role, which privileges the metabolic processes or flows rather than the organism. They see the human being as more like a tube, which facilitates chemical and biological reactions. On another front, since industrialization, the model has shifted from human labor to machine labor, and increasingly in the 20th century, to microbial manufacture, to make food stocks and beverages and fuels and pharmaceuticals. The intent of my project labor then was to paradoxically produce the scent of such human labor as an end product rather than a byproduct or superfluous waste. I wanted my project to reflect industrial society's shift from human and machine labor to increasingly pervasive forms of manufacturing, and in this process to contemplate the changing borders defining what is human. And so I began by capturing my own skin bacteria on pads. And then I harvest the individual colonies before they can be incubated in larger scale fermenting systems. In the current version, I actually purchased the strains from a company called American Tissue Type Collection. But initially, I set out to also isolate the strains from my own body. I concocted for this project special nutrient mixtures that contain sufficient amounts of required sugars while not having much scent of their own. So sweat bacteria can handle lots of salt and mixtures that helps with keeping out opportunistic microbes. In this picture, I'm boiling a nutrient mixture anaerobically for the Propionic bacteria. This is the funkiest smells if it doesn't have oxygen. So the labor project then, this is just the first, showing the first installation of it. It's an installation that recreates the scent of people exerting themselves under stressful conditions, but however, no people are involved. It's created just by bacteria propagating in 3 bioreactors. There are 2 of them in this picture shown on the left and right. The other one's kind of hidden. Sorry to say that I can't reproduce the scent for you here remotely. Just the pictures here. Each bioreactor in the piece incubated then a unique species of human skin bacteria. So Staphylococcus epidermidis, Corynebacterium, and Propionibacterium vitam. And then as these bacteria metabolize simple sugars and fats, they create distinct smells associated with human exertion, stress, and anxiety. The scents in this piece combine in the central chamber, with which a sweatshop icon, the white t-shirt, is infused with scent before they disseminate out. And then the scent increases throughout the exhibition. And I'll read this little bit. I consider the labor installation as a post-anthropocentric scientific theater in which audiences experience a microbial performance and a simultaneous notable absence. In the middle of the room, at the center of the installation, connected to a radiating network of tubes, cables, and bioreactors, is an empty white t-shirt hanging in a glass Belgian. So while humans were at the center of our worldview, or ontology, or epistemology, in labor the human has conspicuous. I say like Robert Boyle's bell jar, at the center of an experimental proof of existence of a vacuum, and the icon of emerging scientific chemistry in the 17th century. This bell jar rests at the center of a new tentative worldview, in which humans have vacated the center. Right? The installation is regulated by controllers that I designed to be like a factory for continuous production. And in this, for me, it was biopolitically important in the Foucauldian sense that the control is not just of laboring bodies, but throughout the process of reproduction, growth and decay, death, fully integrated into the industrial process. In labor, the microorganisms ironically produce the scent of sweat. Not a bulk byproduct, like in the factories of the 19th and 20th century, but as a nostalgic end product. A generative living process as an art installation, or fills the gallery with or toil. I won't maybe get into these, but they're kind of cool. Along with the piece, I created these things called sweat stain transfer prints, which were like they're a novel analog documentation. They're basically full-scale, two-dimensional records of the richly visual temporal moment of sweating, what I consider an unconscious performance. That it was first demonstrated, I first tried it at Cultivamos Cultura in São Luís, in Portugal, in the summer of 2020. And what surprised me about the pictures, I mean, they're made just by essentially taking a wet T-shirt, shaking it up in a bag full of grass, then sandwiching it between two sheets of papers and driving over it with an SUV for pressure. And what was surprising was how they read almost like medical techniques, like or fingerprinting, for that matter. So getting back, and this is the last slide before I show it. While viewers search for visual cues, the sense of labor should create highly charged ambivalence, as audiences semi-human odors created trigger either oppressive or comforting or nostalgic emotional responses. And I hope in this piece that all the clues in the installation allow audience to ponder perverse contemporary ontologies of production with attributes like life inseparable from labor, product and produce, biology as a subfield of technology, and man literally omitted from manufacture. I couldn't have completed this project without the good luck of having laboratory at the University of Buffalo, which the university funded following a competitive proposal for communities of excellence. Coalesce is a hybrid studio laboratory dedicated to enabling hands-on creative engagement with the tools of the life sciences. It's a member of the Environment and Microbiome Community of Excellence. And for me, the labor project reflects on these themes, causation. It's kind of a critique of the genome, looks at the broader causes of who we are, When I put together the specialized tools at the lab for cultivating anaerobic organisms, I somehow thought that there would be lots of labs. What you see in front of you is a cannula system that allows you to basically dose of experiment anaerobically, and also a glove box which we can pump all the oxygen out and have the anaerobic as well. But I realized actually they were very because most of the work into the microbiome was being done in terms of just identifying organisms by their DNA. Like next gen sequencing and other high throughput techniques had allowed science to learn lots about mysterious organisms' DNA, but little about what they ate, or how they metabolize, or how they coexist. So much of what we built a specific growth environment, as well as DNA analysis. My collaborator at Coalesce, Solon, is the lab manager, and he and I put together most of what's in the lab today. Solon is also the scientific collaborator on labor and labor project. And he also works closely with our residents and students working at Coalesce. Typically at Coalesce, there are two graduate students who work in the lab this year. Current students include Daria Warner, shown here, with her fungus. Felipe Shibuya, who's shown here in a workshop with students, and Ryan Hammond. Likewise, each year we also accept 3 to 4 visiting artist residents. In this image, Imam is shown here in a formal residence. From the left, Chris Copeland, faculty member Matt Kenyon, Josh Arthur, and finally John Hopper in the foreground. Here are artists in residence: Sabrina Isatan, working in a research lab. Solon's in the background. And then finally, Nicole Clouston, her large Wingradsky columns in the UV greenhouse, which many of our students, faculty, and residents use each year. We also run several classes each year. This is a course called Biological Art. And the student, in this case, Nate Ellie, produced an amazing series of electron microscope images of diatoms. So he imaged the diatoms on the electron microscope, printed out very enormous iron-like, and then used them for the basis of these cyanotype prints. The guiding mission for me at co-biological art is always meeting goals of my own practice. So Coalesce responds to the grand challenge to our social, cultural, and ethical tools developed through the 20th century by recent advances in design. For instance, what does it mean to design a living organism? What aspects of such an organism could be called an invention or an artwork? How do we presently even define life? Has our reductive worldview and disciplinary isolationism impacted our ability to comprehend the connectedness of life and processes? So Coalesce was built to complement the university's growing sciences by questions the public understanding and participation, but beyond the analytical constraints of most disciplines. That in this image, Van Nguyen, who was the first BioArt TA, so that's basically his Coalesce, just opening up. So I'll conclude and say that for the last 5 years, I've been based in the University Laboratory of Coalesce, dedicated to life sciences branding. But to conclude with some of the assertions I've discussed in my work, I've suggested that genetic codes have little to do with fitness, that race has been constructed in the colonial moment and has been reinforced by reductive DNA hype, that genetic images are essential, that cultural constructs are really built in labs, but that these images are malleable, fakeable, and their authority can be challenged. Furthermore, Raidaki goes as much by non-humans as our DNA. While modern science occupies differentiating, I'm exploring sameness, alikeness, and not difference. I'm not trying to reduce complexity, but rather challenge reductivism. And I've used a strategy taken from Bruno Latour, in which he describes how facts are constructed and authority produced. He describes facts as black boxes. He describes the process of as being strong enough to end the controversy and silence the dissenters. To dispute a fact or to remove black box requires setting up a counter laboratory. And the counter laboratory for me is a place where misleading and colonializing discourse can be refuted. And again, thanks for having me. Everybody's still there.
Yes, we are still here.
Nice.
Just one technical thing. Paul, can you please turn off the microphone of your iPhone?
Oh, I thought it was turned off.
Okay, so that will be fine. Yes. We have a few questions to ask you.
Okay.
So, Professor Chu, who is our curator of this exhibition, would like to ask you to explain how would you differentiate biological experiments and artistic creations?
Yeah, it's a really good question. Having worked in them so long, I see them as creating cultural artifacts. Sorry, I'm trying to move my phone, but I don't think. I see them both as creating these cultural artifacts. And one of the differences that I feel as an artist is I'm trying to stop this. One of the things that I find as an artist is that I'm liberated from some scientific experiments are forced to fit within an existing paradigm, and in a sense, with artwork, we can be consistently defining our own methods. We can be like literally methodological, and through these different methods, we can ask completely different questions that I think can usually be asked. Did you hear me okay?
You were silent for like 5 seconds.
No.
But we can hear you fine now.
Okay. I said basically.
What did you do different?
With artwork, you don't have to be following explicit methods, and one can continually redefine their methods without worrying about staying within a scientific paradigm. Sorry, the echo is distracting, but I think
yes. And Han Ling, who is a Taiwanese bio artist in Netherlands now, and she would like to ask you. There are a lot of debates around the phrase or field of biological art, where some think that it's a movement, and some think you cannot define bio-art, and biological art and bio-art are different things. Some artists even refuse to be labeled as bio-artists, while in Taiwan it's often considered as a subcategory new media art. What would you define your field of work, and how would you comment on the definition of bio-art / biological art? It's a very, very big and long question.
It's a really good question.
Yes, it is.
I've always preferred the term biomedia, because with biomedia I can say I work with biomedia.
Yeah.
But I'm not necessarily saying that the one problem with the term bioart I find is that it defines as this. So it's akin to saying I'm an artist, or I'm a sculpting artist, or I'm a photography artist. So the problem is bioart, labeling oneself as a bioartist, is that it's subcategory, which I don't believe. I prefer the term. I'm an artist who works with biomedia rather than literally bio. However, there's a utility to the term, which is, I think, synonymous with biomedia, and I don't mind that the term does suggest that one working in this form is working with materials from life sciences, connected to living organisms, connected to the natural world, and that if there's a material dimension to any artistic bio material generally related to life and living process. So that's where I sit vis-a-vis that term. I far prefer just to say artists who work with bio. I hope that answers the question.
Yeah, because I remembered in the 2017 manifesto of what is defined as bioart, you specifically said it has to be done with material. So I explain your stance.
Yeah. Well, again, I was trying to. There were some basic things that were at stake in that there was a difference between something made of a material and just a representation. And it was out of a lot of bioart was happening at the kind of end of a postmodern movement where representation and materiality was somehow just seen as a kind of I don't know, a kind of secondary aspect of a thing, right? That culture could be simplified to a kind of symbolic aspect. And the one thing about biomedia, there is something to actual materiality. And I would never suggest that materiality had to be a lab, had to necessarily be by qualities of life. Like, for instance, viruses are not living organisms according to molecular biology. Like I learned, but however, we know they're connected to a deep process of living things. So thus, in the manifesto, it was really just a question of, like, if we're going to label things bioart, right? Mm-hmm. Let's at least have a material. Does that make sense? That there's no sense using a term if it didn't connote something beyond representation.
Mm-hmm. Yeah, and I have a more specific question about your recent work, labor. So I noticed from your website that the bacteria were originally collected from you while you were doing some desk jobs, such as receiving and replying emails in an air-conditioned room. So I'm just wondering, what was your intention of doing instead of collecting these bacteria from, like, workers of a sweatshop or blue-collar laborers or an outdoor work?
Yeah, I mean, originally I used myself because the understanding was that the bacteria was, that this was kind of understanding, and less about the individual and more about these bugs crawling around on us. So the original version was not is not necessarily interested in humans in the way that I think so much of the beginning of the comic era was about. It was all very anthropocentric. In this case, it didn't really matter for me who the bacteria came from, because it's much more than it was human, right? I was really trying to show the vacated space of humanness, not like the sort of specifics of located work. So when I was trying to put myself through different situations of sweating in different ways, it was merely to try to the proper types of bacteria related not only to physical activity, but also to kind of a nervous or stressful activity.
Yeah. Sorry.
I will say one other thing is also at the end, I wound up realizing that in terms of making the work something that could also be, I guess, friendly to museum spaces or somehow move global, somehow be taken into museum spaces. I got into the idea of sometimes purchasing the species online. And one of the reasons I did that was I wanted to know exactly what was in the different the bioreactors. I didn't want this to be a piece where there was this ambiguity about what was in there. I needed it to be safe for the public and be understood by the institutions that hosted it.
Yeah. And so another extended question from the biosafety thing is that, does your perspective of your work or artwork in general changed by the COVID-19 pandemic at all?
Yeah, completely. I'm still figuring out how. I mean, I think this whole notion of post-anthropocentrism that the labor piece was dealing with is certainly applicable, right? This idea that we've been so fixated on ourselves that we've found it nearly impossible to consider the other kind, that we exist also still in a broad connected ecosystem. Which doesn't necessarily see us or even our time frames as central. It doesn't see our economic systems as somehow better than life. So in this sense, I feel like the labor piece was, in a sense, a bit prophetic in some ways. Well, it had me thinking about it. But one of the things I've noticed was that this idea of virus and virus as metaphor is something that doesn't show up in a lot of biomedia work, which I think is intriguing. And I thought about the reason why, and I think one of the reasons why is that the metaphor of virus was so prevalent in the 1990s because of computer virus and the fact that computer virus could be seen as analogous to real virus. I think there was a kind of reluctance by a lot of, biomedia artists to enter into that space again, because again, they were metaphors made so familiar in the 1990s. Does that make sense?
Yes. Do you think it's important for like emerging media artists to do projects related to emerging media itself?
Wait, say that again.
I noticed that you start working with DNA materials the end of the Hugo project, so sort of post-genome era. And do you think, and I heard your opinions about the committing and related artwork. And I'm just wondering, do you think it's important for artists who work with emerging media to do projects related to the media itself? So, I mean, to do projects discusses with the current issues or disputes related to the media itself.
Yeah, I think it's important to understand the way in which media signifies, right? Mm-hmm. Part of the reason I talk about working self-reflexively is because, especially when one's working with new technology, the first thing someone sees is the medium itself. This is kind of like a Marshall McLuhan, right? That the medium is the message. That like when people in the 1970s turned on a TV, the first thing they saw was TV, the TV signal, and secondly they saw the actual content which was being delivered, right? That the media itself always has a message, whether it's always a subtext that is communicated through it. And I'm particularly interested in emerging forms that are like so new we're not used to them communicating, because I feel like they automatically bring that subtext right in front of us, right? And if anything, we're not expecting that form to communicate in the way that traditional media does. And so for me, it's the idea of using new media. The whole challenge is forcing that medium to say something, right? It automatically is something. It has this presence. It has this kind of being itself. But then trying to make that also communicate with a kind of nuance, I think is, I guess, the very interesting challenge for artists. So I wouldn't say every artist that works with a form has to, must address that form. But for me, that's the interesting challenge. And I still think a lot of what making art is pushing yourself and challenging yourself to say something that hasn't been said. And for me, a lot of that saying has to do with kind of creating the kind of proper kind of poetic relationship between the sort of form and what is communicated.
So someone anonymous wants to ask you, have you ever faced difficulty to communicate with your audiences? And how do you make sure that audiences can understand what you are trying to express?
What I've always tried to do is I've always tried to communicate literally as clearly as possible. Okay. but mostly using nonverbal means.
Okay.
So in all these works, I feel like mostly what I do is I work and I work and I work to try to make everything as literally as clear as possible. I never am trying to produce, never trying to confuse. Okay. I'm always trying to say very directly, but the media itself, because it's not a typical communication medium, doesn't always, it's not something people always recognize, right? Like, I really wanted people to understand how gel electrophoresis worked when I did the DNA work. I was never trying to hide it. I was doing it in public. I was explaining it. Half the time my performances were just explaining how it worked. I was doing everything I possibly could to kind of allow the medium to communicate. Although sometimes, whether it's the media itself, the medium that I chose itself, that isn't fully understandable for everybody. And sometimes it's simply also a matter of the people's backgrounds are never as perfect as you want. I will say that ideally I really like the writing of Jorge Luis Borges, who's a science fiction writer from Argentina. He was born about 100 years ago. But I really like the writing, but it does presume that an audience is like really fascinated, paying attention, and also it kind of assumes that an audience will sometimes read the book and then want to go to the library and read the sources. So I've always recognized that I've created something complex for people to understand, but I've never created complexity just for the sake of complexity. I've always tried to make it clear and as concise as possible.
Mm-hmm. And so someone anonymous also wants to know about your creative strategy and how you transform. How do you usually transform bio tissues or biomaterials to become artistic materials?
I think a lot of times my process begins by just seeing something and then thinking about what the opposite of that would be. Literally, just a very simple technique. So when I first saw a DNA fingerprint and somebody said, "Oh, that's your genetic portrait. That's the real you. That's who you really are," I thought, well, what if it's not? And what if it's not? What if how is that thing made? And maybe this isn't as authoritative as everybody is telling me about. And so it begins with a kind of a playful negation and to be open to the idea that something isn't true or that something can be reversed. So with the DNA image, I just began with the idea, what if I could make pictures of that? With it, what if I could control it? And then thinking through that, then you all of a sudden realize, wait, but that might be kind of profound, because it wouldn't just be making a picture, it would be using something which is normally used as a proof or as an evidence to become a kind of artistic whimsy. So for me, it usually begins with a playful negation of something that might be held obvious.
And so have you ever encountered any ethical issues during your exhibition or while preparing for your artwork?
Yeah,
what do you think of ethical issues? Do you think art should take ethical issues into account?
Absolutely. I mean, I think that's the thing that we kind of have to be doing. It's the thing which hasn't been supported society very well. I think there's been a lot of societal effort on developing new technologies, and not nearly enough updating our ethical sensitivities. I've been critical. Well, I guess with labor, one of the things that I do is I work with native organisms, like literally organisms that have not been genetically modified.
Yeah.
And I do this partially because there's a way in which synthetic biology has become a kind of default way that we think about organisms. That, okay, if we're going to work with an organism, the first thing we have to do is transform its genome. We have to radically genetically modify this organism. And I don't take that as a given. I think there are all kinds of different ways in life, and many of them actually involve understanding the way an organism works in the naturally, right? Or the way an organism works that has evolved, without human intervention. One of the I think the largest issues that we're facing as a kind of, if we think of biomedia as a kind of movement, we want to buy into the sort of like the imperative of genetic engineering. I've resisted it for various reasons. Part is that I don't want to be just the default way we all work, because I think many of the tools of genetic engineering have tried to become like Lego building blocks, and it's tried to become kind of almost like Photoshop software toolkit for how we're supposed to proceed as artists. That the goal of bio art is somehow to make modified organism that's our own creation, and I don't buy that. I think there are hundreds of thousands of other ways of thinking about what we do as artists. I find a lot of synthetic biology to be, in a sense, playing by certain kind of biotechnology rules. Don't go too closely. Does that make sense?
Yes. And do you think it would be different if, like, for example, labor, the American project, is exhibited in a science gallery or a science museum instead of an art gallery?
You know, I would say if you asked me that 15 years ago, I would have said I'm not interested in that. I find that the context of the Science Museum makes us focus on the wrong thing, would make the message never possibly be. We're always privileged to kind of de-whiz over the a more kind of thoughtful engagement. But I think at this point, I think the modern science museum has changed. I think the way that it's like natural history in Vienna, or the science gallery in England, or all these different science museums, they're now talking to people in a very different way. And I think I think in a way they are doing a better job of basically, integrating the arts, integrating criticality, creating, allowing for a kind of viewer that is not simply there for the kind of gee whiz. And so at this point, no, I actually feel like science.
Yeah.
These are great questions, by the way.
I think I agree with you. And there's one more question upcoming. And it has to be the last question because we are running out of time. So Yanting Guang, who used to work in the National Art Gallery, and she wants to ask you, according to your practices and experiences, do you think bioart is cultural, that it is still significant when you read it in different cultural contexts?
I can't always know the answer to that. I mean, I do come from a specific cultural place, so I can't always answer to what everyone's experiences are. Exactly. The idea is that when you're working with symbols and representation in these ways, it's like you never know exactly how someone reads that. So I can't know for sure. I also feel like I try to make these works like that. There's no text. There's no. I've tried to move away from any kind of explicit language in my work. However, I also feel like the issues that I'm dealing with very well might be ones that are best understood in certain parts of the world than others. and I find that unfortunate. I feel like I can't necessarily transcend my own cultural place and attempt to make an art which is necessarily universal. But I certainly try to make it something that is open, that doesn't require specific verbal, even if it might still have a kind of idealized viewer in mind that somehow is conversant and is also politically situated to kind of understand the kind of ethical dilemma of which they're a part. I hope that's
I think it's quite interesting talking to you, and we are all looking forward to see you in Taiwan, and so we can talk more in more greater details. So about time is up. So thank you for joining us online from New York, and we will see next year in Taiwan, hopefully.
Yeah, nice. Yeah, looking forward to it. Again, thanks for all the questions.
Take care of your health. Take care.
Appreciate it. Bye-bye.